Now look at this.
What's up, Hippie Pie Penguins? Today we're going to do topic 7.4 on population genetics. So first thing, what's a mutation? So a mutation is going to be a change in the DNA. And if you change the DNA, there's a chance that the protein is going to change. And so if you change the protein, then you're going to see a change in your phenotype. Natural selection is going to act on these different phenotypes. So as we have different variety in our population, and as the environment constantly changes, certain traits will be more favorable and other traits will be less favorable. So we want there to be a lot of genetic variation within our population. Problem is, genetic drift is going to affect this. Genetic drift is going to be different random situations that happen. There are two different types of genetic drift. You have bottleneck effect and you have the founder effect. Bottleneck effect is going to be a random situation that takes place that drastically decreases your population size. Now, the new population after the event is not going to be representative of the original population. So the allele frequencies have changed, and that is going to lead into evolution. So this picture kind of gives you a better representation of the changes in that allele frequency, so you can kind of see how the original population and the new population do not have the similar allele frequencies. So in founder effect, this is where we're going to see a small set of the population gets isolated or separated. So it's kind of like if you take your hand into a bowl of alleles and you just plop them somewhere else. It's random which of the organisms are going to become isolated. It's random which of the organisms kind of go to the new area and form this new population. And again, the new population is not going to be representative of the original population. And so those allele frequencies are going to be different. Gene flow can also affect our allele frequencies. Gene flow has to do with immigration, where individuals are moving into the population. Emigration is where individuals are moving out. So I always remember like the E for exit and the I for in. And so with this is going to bring new alleles into our population, or could cause certain alleles to leave our population. And that affects our allele frequencies. Also, an increase in gene flow is going to decrease the genetic variation between populations. So natural selection, you usually kind of see three different things. You'll see directional selection, stabilizing selection, or disruptive selection. Directional selection is going to move in a direction toward a certain phenotype. Stabilizing selection kind of reinforces the intermediate, and disruptive selection kind of gets rid of the intermediate, and we see the two extremes increase. So in directional selection, we see it's moving toward a certain extreme or moving in a direction. The peppered moths are a great example of this. Peppered moths used to be lighter in shade, then the Industrial Revolution darkened the trees. So the individuals that were darker were more favorable. So we saw that the population shifted toward the darker phenotype. Stabilizing selection is going to be where we're reinforcing that intermediate. So you kind of find that most babies are born in that 6 to 8 pound range because of stabilizing selection. With the rock pocket mouse, we see that there's going to be disruptive selection. So you have the light ones that are more favorable in the sandy environment and the dark ones that are more favorable in that lava area, and a decrease in intermediate. API Penguins just says, bye y'all.